welcome back to Kitchen Science Biology Lab with Professor Tracy. Right now we're just going to do a really quick demonstration here using goldfish crackers as alleles to map out what exactly happens during a monohybrid cross. This is one way we can study Mendel's law of segregation when we're looking at genetics and predicting the future offspring that might happen when we have two parents reproducing. So in this case, what we're going to do with our law of segregation is that we're going to say this was our phenotype, or sorry, our genotype of our parents. That both mom and dad were heterozygotes, so they had one copy of the red allele and one copy of the orange allele. We go through meiosis, and half of our gametes are going to have this allele, and half of our gametes are going to have that allele. To keep things simple, rather than drawing a 4x4 square, we're just going to do a 2x2 because half of those 4 gametes were red and half of those 4 gametes were orange. And so we're just going to make it a little bit simpler here by putting down one representative gamete of each type. So when we're doing a Punnett square, what we need to do first is label, mom's alleles and dad's alleles here. So they both have the same genetic makeup, one red, one orange. And then what we're going to do next is we're going to copy this information down to these two potential kids. So mom is donating a red allele to those two potential offspring. And on this side, this gamete is going to donate a orange allele to those potential gametes. And we're going to do the same thing for dad. So dad's going to be able to donate with his sperm cell a red allele to both this offspring and this offspring. And going across the bottom, dad is donating an orange allele to both this offspring and this offspring. And so now we have our 4 potential kids here, genetic combinations, based on mom being a heterozygote and dad being a heterozygote. So what does this mean for what we call the genotype? Recall that the genotype is going to be the letters that we see. So in this case, we have one of our little squares of offspring has a combination of red, red. So that represents 1 out of 4 offspring have the big R combination. We look at the next one over. This offspring is a heterozygote just like the parents. 1 orange, 1 red. 1 orange, 1 red. So in this case, we have big R, little r, and we have 2 out of our 4 cells have this combination. We can reduce that fraction down. Instead of writing 2/4, we're going to say half of our potential offspring in this 4 grids has the heterozygote condition. And then lastly, down here in this bottom corner, we have our homozygous recessive of little r, or orange, orange. So we've done one Punnett square to quickly represent what would happen if our parents were both heterozygotes. Let's switch it up. In this case, let's say instead of mom being a heterozygote, we're going to make mom homozygous dominant. And just because we already have the 4 lines drawn, we're going to keep it the same. So mom, again, donating a red to these 2 offspring, and then also mom is donating a red to these 2 offspring here. Now we're going to go across the row. Dad is donating a red to both of these offspring, and over here dad is donating an orange to these offspring during fertilization. But how does this mess up our ratios? When we had both heterozygotes, we predicted, or we showed, that there were 1/4 big R, 1/2 big R, little r, and 1/4 little r, little r. But now that our combination is big R from mom and big, little from dad, what does this do to our ratios? Well, now we look at our potential offspring. This offspring has big R, or red, red. This one also has the same thing. So now 2 out of 4 have big R. So half of our offspring are now homozygous dominant. Our other 2 offspring are both heterozygotes, big R, little r. What does this do to our phenotype ratios? In our first cross, we had 3/4 were going to be red and 1/4 were orange. In our second cross here, we come up with a different ratio. That we have all of our offspring are red because we do not see any of our little r, little r. None of our 4 cells have 2 recessive traits. So when we use a Punnett square like this, we can use our Punnett square to quickly map out what will the offspring look like. And this is following Mendel's law of segregation, in that each gamete needs to be represented by that trait. If we were to, say, have an error in meiosis where we do not have a second allele from mom, then we missed out. Something happened. We're not going to be able to produce a successful offspring in this case because our gamete was missing this particular trait. If this trait was essential for the fish's livelihood, maybe in this case, maybe it would be albino because it's lacking pigment on that allele. But if we have something essential like this is how the heart muscles are forming, and that's missing, we're not going to have a successful production of a gamete that can result in any offspring. So when we're looking at a Mendelian traits telling us, Mendelian laws, that each gamete needs to be representing each individual trait or allele that's present in our DNA. Stay tuned for a dihybrid cross.